Hi friends, welcome back to Anand Science Academy. Let's continue the chapter Principles of Inheritance and Variation. In the previous video, we have learnt what are the 7 characters and their contrasting traits studied by Mendel in Pisum sativum plant. In this video, let's understand how he carried out the hybridization experiments and what are the steps involved in the hybridization process in a self-pollinated plant like Pisum sativum. The first step in hybridization experiment is selection of parents. Mendel selected a tall pea plant and a dwarf pea plant. Both were pure breeding lines. Already we have learnt what is a pure breeding line. Hope you remember that. The second step is emasculation. It's an important step. Why this emasculation to be done? Let us see. At a certain stage of flower development, the anthers become ripe and dehisce to liberate the pollens. This is known as anthesis. These anthers must be removed before anthesis from the flowers of the female parent to prevent self-pollination. This removal of anthers is known as emasculation. This step is required only in a bisexual and self-pollinated flower. Pea plant has bisexual flowers which are self-pollinated. So, this emasculation step is crucial in the hybridization experiment. The next step is bagging. This is done after emasculation. Bagging is the protection of emasculated flower from contamination by undesirable pollen grains, because by wind the pollen grains from other plants may come and fall on the stigma, so it may lead to some unwanted pollination. To prevent that, bagging is done. This is done after emasculation. The emasculated flowers should be kept properly covered so that no untoward cross-pollination takes place. This is done by enclosing the emasculated flower in a muslin cloth, oil paper or polythene bags. So, this step is done to prevent contamination by undesirable or unwanted pollen grains. The next step is pollination. Here one thing we have to note down. This is controlled pollination, not natural pollination process. Because to avoid that natural self-pollination only, we have emasculated the bisexual flower and we have bagged the flowers to prevent the unwanted pollination. So here we call it as controlled pollination. When the stigma of the emasculated flower becomes mature or receptive, we call it as, it is artificially pollinated by the pollens taken from a male plant or male parent. During pollination, whole freshly dehisced anthers are plucked from the flowers of the male parent by means of forceps. These forceps should be sterilized. And these pollen grains from these anthers are dusted on the stigma. The bags are temporarily removed at the time of pollination and again it is replaced as before and after pollination. Let us just recall what are all the steps we have seen now. First one, selection of parents. Which plant I am going to take as a male parent, the other one as a female parent, that is selected. Next, emasculation, removal of anthers to avoid self-pollination. Next one, bagging. Until the stigma becomes receptive or mature, the flowers, emasculated flowers are bagged, covered to prevent contamination by other pollen grains. The next process is pollination or controlled pollination. Anthers from the desired male parent are collected and dusted on the stigma artificially. So pollination gets over. Let's see what is the next step. The next step is tagging. This is also very important. The crossed flowers should always be kept properly tagged or labeled. The tag should be brief but complete. The tag should bear the names of the parents. The name of the female parent should be written first. It must contain a number referring to the field notebook. All necessary particulars about the cross should be recorded in the field notebook under that number, so that it will be easy for us to study what is the male parent, which is the female parent, and when this emasculation was done, when this pollination was done. All those details will be there in the tag. The last step in hybridization experiment is selection. The seeds are allowed to mature within the bags. Then they are harvested and stored with proper labeling. From the next growing season onwards, the hybrid seeds are grown and they are allowed to self-pollinate, and the selection is carried on to find out the desired phenotypes and make them homozygous. Let's take the example of one such hybridization experiment carried out by Mendel, where he crossed tall and dwarf pea plants to study the inheritance of one gene, that is, the gene responsible for the height of the pea plant. Here this picture shows how he carried out the cross-pollination or hybridization and what are all the results he observed. He collected the seeds produced as a result of this cross and grew them to generate plants of the first hybrid generation. This generation is also called the filial 1 progeny or the F1 generation. Mendel observed that all the F1 progeny plants were tall, like one of its parents. None were dwarf. Here you have to understand this. Mendel crossed a male tall plant and a female dwarf plant. Again, he conducted the experiment vice versa also, and he got the F1 progeny all tall plants. That means one of the parental characters is expressed in the F1 progeny. Mendel made similar observation for other pair of traits also. He found that the F1 generation always resembled either one of the parents, and that the trait of the other parent was not seen in the F1 generation. Say for example, here the picture shows that a plant with violet flowers and white flowers were crossed, but in the F1 progeny all the flowers were purple or violet in color. One of the parental character is expressed. Hope you are clear with this, friends. Mendel crossed a tall plant and a dwarf plant. In the F1 generation, all the plants were tall. This is one process, one step over. What is the next step in Mendel's experiment? Mendel self-pollinated the tall F1 plants to observe the inheritance of characters. To his surprise, he found that the filial 2 generation or F2 generation, some of the offspring were dwarf. The character that was not seen in the F1 generation was now expressed. The proportion of plants that were dwarf were 1/4 of the F2 plants, while 3/4 of the F2 plants were tall. This picture shows it clearly. First, parental generation, tall plant and dwarf plants were crossed. In the F1 generation, all the plants were tall. These F1 plants were allowed to self-pollinate, as a result of which the progeny produced were 75% tall and 25% dwarf. Hope you got it. The tall and dwarf traits were identical to their parental type and did not show any blending. That is, all the offspring were either tall or dwarf. None were of an in-between height. Similar results were obtained with the other traits that he studied. Only one of the parental traits was expressed in the F1 generation, while at the F2 generation, both the traits were expressed in the proportion of 3:1. The contrasting traits did not show any blending, mixing or in-between character either in F1 or F2 stage. Based on these observations, Mendel proposed that something was being stably passed down, unchanged from parent to offspring through the gametes over successive generations. He called these things as factors. Now we call them as genes. Genes, therefore, are the units of inheritance. They contain the information that is required to express a particular trait in an organism. Genes which code for a pair of contrasting traits are known as alleles. That is, they are slightly different forms of the same gene. Alleles are the slightly different forms of the same gene. If we use alphabetical symbols for each gene, then the capital letter is used for the trait expressed at the F1 stage or F1 generation and the small alphabet for the other trait. For example, in case of the character of height, capital T is used for the tall trait and small t is used for the dwarf, and capital T and small t are the alleles of each other. Hence, in the plants, the pair of alleles for height would be capital T, capital T, capital T, small t, or small t, small t. Mendel also proposed that in a true breeding, tall or dwarf P variety, the allelic pair of genes for height are identical. This condition is known as homozygous condition. Say for example, if the allelic pair for tall plants is capital T and capital T, it is known as homozygous tall. If it is a dwarf plant, the allelic pair for the expression of this character is denoted as small t, small t. This is also homozygous condition. Capital T and small t are called the genotype of the plant, while the descriptive terms tall and dwarf, purple, white, round, yellow, such descriptions are known as phenotype. Genotype is the pair of alleles present in the plant. Phenotype is the appearance or expression of the character. Can you answer this question? What then would be the phenotype of a plant that had a genotype capital T and small t? Yes, you got it right. It is tall. As Mendel found the phenotype of the F1 heterozygote capital T small t to be exactly like the capital T parent in appearance, he proposed that in a pair of dissimilar factors, one dominates the other and hence is called the dominant factor, while the other factor is recessive. In this case, capital T for tallness is dominant over small t for dwarfness, that is recessive. He observed identical behavior for all the other characters or trait pairs that he studied. It is convenient to use the capital and lowercase of an alphabetical symbol to remember this concept of dominance and recessiveness. It's better not to use capital T for tall and D for dwarf because you will find it difficult to remember whether capital T and small d are alleles of the same gene or not. Alleles can be similar as in the case of homozygotes capital T and small t, or can be dissimilar as in the case of heterozygote capital T and small t. Since capital T small t plant is heterozygous for genes controlling one character, it is a monohybrid and the cross between capital T and small t is a monohybrid cross. From the observation that the recessive parental trait is expressed without any blending in the F2 generation, we can infer that when the tall and dwarf plant produce gametes by the process of meiosis, the alleles of the parental pair segregate or separate from each other and only one allele is transmitted to a gamete. This segregation of alleles is a random process, and so there is a 50% chance of a gamete containing either allele, as has been verified by the results of the crossings. In this way, the gametes of the tall capital T plants have the allele capital T, and the gametes of the dwarf small t plants have the allele small t. During fertilization, the two alleles capital T from one parent, say through the pollen, and the small t from the other parent, then through the egg, are united to produce zygotes that have one capital T allele and one small t allele. In other words, the hybrids have capital T, small t. Since these hybrids contain alleles which express contrasting traits, the plants are heterozygous. The production of gametes by the parents, the formation of zygotes, the F1 and the F2 plants can be understood from a diagram called Punnett square or checkerboard, we call it as. It was developed by a British geneticist, Reginald C. Punnett. It's a graphical representation to calculate the probability of all possible genotype of offspring in a genetic cross. The possible genotypes are written on two sides, usually the top row and left columns. All possible combinations are represented in boxes below in the squares, which generates a square output form. The right side of this slide, you can see a sample Punnett square. The Punnett square shows the parental tall, capital T, and dwarf, small t plants. The gametes produced by them and the F1 progeny, that is capital T, small t. The F1 plants of genotype capital T, small t are self-pollinated. The symbols to denote male and female also you can see at the top. The F1 plant of genotype capital T, small t, when self-pollinated, produces gametes of the genotype capital T and small t in equal proportion. When fertilization takes place, the pollen grains of genotype capital T have a 50% chance to pollinate eggs of the genotype capital T as well as of genotype small t. Also, pollen grains of genotype small t have a 50% chance of pollinating eggs of genotype capital T as well as of genotype small t. As a result of random fertilization, the resultant zygotes can be of the genotypes capital T, capital T small t or small t. You can see this represented in the Punnett square along with the phenotypes. From the Punnett square, it is easily seen that 1/4 of the random fertilizations lead to capital T and 50% or half of the proportion lead to capital T small t and 1/4 to small t. Though the F1 have a genotype of capital T small t, but the phenotypic character seen is tall. At F2, 3/4 of the plants are tall, where some of them are capital T, while others are capital T small t. Externally, it is not possible to distinguish between the plants with the genotypes capital T and capital T small t. Hence, within the genotypic pair capital T small t, only one character capital T tall is expressed. Hence, the character capital T or tall is said to be dominate over the other allele small t or dwarf character. It is thus due to this dominance of one character over the other that all the F1 are tall, though the genotype is capital T small t. And in the F2 generation, 3/4 of the plants are tall, though genotypically half are capital T, small t, and only 1/4 are capital T, capital T. This leads to a phenotypic ratio of 3/4 tall is to 1/4 dwarf. That is 3:1 ratio. But a genotypic ratio of 1:2:1. Hope you are able to understand, my dear friends. The 1/4 is to 1/2 is to 1/4 ratio of capital T, capital T, capital T, small t, small t is mathematically condensable to the form of a binomial expression ax plus by whole square that has the gametes bearing genes capital T or small t in equal frequency of half or 1/2. The expression is expanded as given below. Mendel self-pollinated the F2 plants and found that dwarf F2 plants continued to generate dwarf plants in F3 and F4 generations. He concluded that the genotype of the dwarfs was homozygous, that is small t, small t. What do you think he would have got if he had self-pollinated a tall F2 plant? Can you guess? If the homozygous tall plants are self-pollinated, they produce gametes with an allele capital T. So, the progeny produced will be all tall with the genotype of capital T, capital T. That means all homozygous tall plants. If the heterozygous tall plants are self-pollinated, they produce two different kinds of gametes, either with capital T or with small t, and the progeny will be both tall and dwarf in the ratio of 3:1. That is phenotypic ratio. And the genotype will be 1:2:1. From the preceding paragraphs, it is clear that though the genotypic ratios can be calculated using mathematical probability, by simply looking at the phenotype of a dominant trait, it is not possible to know the genotypic composition. That is, for example, whether a tall plant from F1 or F2 generation has capital T or capital T small t composition. It cannot be predicted. Therefore, to determine the genotype of a tall plant at F2, Mendel crossed the tall plant from F2 with a dwarf plant. This he called a test cross. In a typical test cross, an organism showing a dominant phenotype is crossed with the recessive parent instead of self-crossing. The progenies of such a cross can easily be analyzed to predict the genotype of the test organism. The figure given here shows the result of typical test cross where violet color flower is dominant over white color flower. The alleles are represented as capital W and small w. You need not get confused with this W and W, capital W and small w. You may think violet is a dominant trait so that we can have capital V and small v. Yes, in that way also you can take. But as it is given in the NCERT reader, I am using the same alphabets here. Using a Punnett square, try to find out the nature of offspring of a test cross. What ratio did you get? Using the genotypes of this cross, can you give a general definition for a test cross? Yes, it's possible. If the given violet colored flower is homozygous for the character, that means it has two dominant alleles. Say here capital W, capital W. It is crossed with a white colored plant with alleles small w, small w. The F1 progeny produced will all be violet colored flowers. 100% violet flowers will be produced. If the violet flower producing plant is heterozygous for the character, that means it has one dominant allele and another recessive allele. Here capital W and small w. It is crossed with the white colored plant. Then in F1 progeny, the ratio of violet and white flowers will be 1:1. Can you follow this? If the violet color flower bearing plant is homozygous, when it is crossed with white colored flowers, all the F1 progeny will have violet color flowers. But if it is heterozygous for the trait, when it is crossed with the recessive or white flowers here, the progeny will have violet and white flowers in the ratio of 1:1. Based on his observations on monohybrid crosses, Mendel proposed two general rules to consolidate his understanding of inheritance in monohybrid crosses. Today, these rules are called the principles or laws of inheritance. The first law or law of dominance and the second law or law of segregation. This law is also known as law of purity of gametes. Let's see what is this law of dominance. According to this law, characters are controlled by discrete units called factors. Factors occur in pairs. In a dissimilar pair of factors, one member of the pair dominates the other. The law of dominance is used to explain the expression of only one of the parental characters in a monohybrid cross in the F1 generation and the expression of both, that is dominant and recessive character, in the F2 generation. It also explains the proportion of 3:1 ratio obtained at the F2 generation, that is the phenotypic ratio. The second law, law of segregation or law of purity of gametes. This law is based on the fact that the alleles do not show any blending and that both the characters are recovered as such in the F2 generation, though one of these is not seen at the F1 generation. Though the parents contain two alleles, during gamete formation, the factors of alleles of a pair segregate from each other such that a gamete receives only one of the two factors. Of course, a homozygous parent produces all gametes that are similar, while a heterozygous one produces two kinds of gametes, each having one allele with equal proportion. We can have the definition for law of segregation like this. The two alleles of a gene separate during formation of gametes. They do not blend but segregate or separate into different gametes. Hence, each gamete is pure and possesses only one allele of a gene. This definition is not given in our NCERT text, so that you can use this definition for this law of segregation. Okay, my dear friends, hope you are all clear with what we have discussed in this video. Let's continue this chapter in the next video. Good luck.